Hi, this is Dr. B, and today we're going to talk about meiosis. But first, let's do some comparisons between mitosis and meiosis. So we talked about mitosis in the last video that we did together. At the end of each cell, it has 2 copies of a chromosome. It makes exact copies. It's what your skin does to replace your skin because you're always having skin flaking off. It happens in boring places like your toes and your fingers, your eyeballs, and places where skin cells are always dying off, or cells in general are always dying off and need to be replaced. It also happens in growing people like toddlers and teenagers who are growing quickly. It happens in almost all body cells because we need to replace our cells on a pretty regular basis. It's less complicated than meiosis, which is what we're going to study today. Mitosis produces body cells. They're cells that are not for reproduction. Mitosis is a cycle, and in other words, it creates cells that can create more cells. Meiosis is not a cycle. Meiosis is a process where crossover happens. That's where genetic diversity occurs. And we'll talk about that when we look at meiosis. We're going to look at a meiosis simulation here. At the end, each cell has 1 copy of a chromosome. That's different than mitosis because mitosis has 2, meiosis has 1. Meiosis is for sexual reproduction, so it's eggs and sperm. It's not a cycle because it ends at those eggs and sperm, and then they have to find eggs have to find sperm, sperm have to find eggs to get 2 copies of the chromosome again and keep going. So it's not a cycle, it's a stop and find a partner kind of process. It happens only in reproductive tissue, so it's a little bit different than mitosis that happens everywhere. It's more complicated because you have to go through cytokinesis twice, and you have to line up all the chromosomes twice to get everything split. So each cell has 1 copy of the chromosome. It produces eggs and sperm, and like we said, it's a stop in the name of love. It's not a cycle. It has to find a partner cell to continue. Here's a simulation we're going to be using in Gizmos. It's a simulation of meiosis, a type of cell division in which the parent cell being a cell in your body that produces reproductive cells, called like a seed cell. It produces 4 daughter cells with half the number of chromosomes at the parent cell. Excuse me, it's a germ cell. Choose a male or female parent cell to begin with. Well, since females do all their meiosis before they're born, and they do meiosis first, let's start with them. So let's do female meiosis. The cell grows. The germ cell, the cell in your body that creates eggs, or the cell in the female body that creates eggs, doubles its DNA. So we're copying our DNA here. It's doubled. We're in prophase 1 here. Here's a little map of all the different phases of meiosis. After the DNA is copied, the cell grows more. Click on the nucleus to break it down. We're breaking down the nuclear envelope, the nuclear membrane, so the chromosomes can spread around the cell and be eventually lined up to divide. So we're going to condense those chromosomes so it's easy for them to not get tangled up as they divide. That's what your cell does. We're dragging the centrosomes. They're kind of organelles that help organize the chromosomes as the cell splits. Homologous chromosomes, remember, are chromosomes that contain the same genes but not necessarily the same alleles. So let's say this is chromosome number 1 and this is chromosome number 2. There's 2 sets there. We're going to do what's called crossover here, where the 2 chromosomes, one from the mother, one from the father, that previously haven't mixed in the organism yet, have an opportunity to mix up their alleles here. So now we've made 1 crossover per chromatid, and let's hit continue here. So we're going to drag the homologous pairs. They're in what's called a tetrad. Tetrad stands for 4. So there's 4 copies of each chromosome here on this this central area here. And then we're going to drag spindle fibers from the centrosome here. And this is going to help everything stay tidy as the cell divides. We're going to shorten the spindle fibers here, and you can see how the spindle fibers kind of pull the chromosomes to each side of the cell. And then the nucleus is going to reform. We're going to squish the cell in half, called cytokinesis. So that's our first cytokinesis. Cytokinesis means cell movement. Cyto means cell, and kinesis means movement. So now we need to do this again. We need to go into phase 2 because now each cell here has 2 copies of the chromosome. In order to get to sex cells, we need to get down to 1 copy each. So when they get with a sperm, they have 2 copies, and only 2. Because if you had 2 and then 2, you get to 4 and things would get messed up. So the whole process of meiosis is to get down to 1 copy in each of those reproductive cells. So we're going to reorganize these as tightly bound chromosomes. We're going to get the centrosomes ready to go here. They're only doing right and left here to help you see that this is different. This is Metaphase II. And this time, instead of lining up as tetrads, they're just lining up as identical chromatids here, sister chromatids. Well, actually, they're not sister chromatids. They're chromatids. They've done crossovers, so they're not sister chromatids. So you can see that the chromosomes are not identical here. So this is one of the places that randomness is introduced. If this chromosome were flipped, or if this one were flipped, you'd get a different mix of alleles here. Same thing with the purple chromosome here. So we're going to drag spindle fibers here to just keep everything organized and make sure the chromosomes get where they're going. The cell has a lot of organizing to do. They only show 2 chromosomes here, but in humans there are 23. In cats there are 19. Every organism, many organisms have different numbers of chromosomes. So we're going to click on each chromosome here, or excuse me, centrosome. And now you notice there's only 1 copy of each chromosome in half of the cell here. We're going to reform the nuclei. And we're going to drag the cells apart. Now in females, you get 1 egg cell and you get 3 polar bodies. So you get 4 cells total, but only 1's going to turn into the egg. These polar bodies are kind of just recycled. So we're going to click here. This egg cell will mature and specialize a bit here. The coating around it will develop a bit, and then it will be a mature egg cell and go on to perhaps be part of reproduction. We're going to reset, and we're going to look at meiosis in males. It's pretty much the same, but at the end things are a bit different. But it gives us an opportunity to again look at some of the randomness that gets introduced, some of the variation that happens when meiosis happens. So we're going to double the DNA. Remember, we're in prophase 1. This is a germ cell, the cells that create eggs and sperm. So we're doubling that chromosomes. So now we have 4 copies. So you can see we have 1, 2, 3, 4 of the green chromosome and 1, 2, 3, 4 of the purple chromosome. The purple chromosome here is a sister chromatid. These are exactly the same right now. We're going to drag our centrosomes here. We're going to pair them up here. And we're pairing up to make a tetrad here. This group of 4 chromosomes is a tetrad. And this is where crossover happens. What was the 2 parent chromosomes that haven't crossed over in the organism yet, that haven't mixed, now get an opportunity to mix here and crossover. So some of the alleles get swapped and we made new chromosomes. So crossover is one of the opportunities where randomness or variation can occur. So now we can continue. We line up our tetrads on the metaphase plate, and notice in metaphase 1, we've got tetrads here in the middle. We're going to drag our spindle fibers here down to just keep everybody really nice and organized. The cell's got a lot of work to do in organizing all the chromosomes and making sure everything goes in the right place. So now we're in anaphase 1. We're going to pull things to opposite poles, get organized here, so we can split the cell in half. We're going to reform the nucleus. This doesn't happen in every kind of cell. You don't always reform the nucleus. Now we're going to recondense here, make our chromatids, drag our centrosomes. Now here's another opportunity for randomness. We're going to line these up on the metaphase plate, and because we've done crossover now, and because these are homologous chromosomes, they're not exactly the same. And so when that divides, anytime you line up cells on the metaphase plate, during metaphase 1, metaphase 2, you're going to get an opportunity for randomness to happen. And you have opportunities for gametes with different alleles to occur. Remember, alleles are versions of a gene. Everybody's got the same genes. We all have a gene for, say, the size of your nose, size and shape of your nose. And what allele you get for the size and shape of your nose, it's actually probably several genes, but let's just, for example, say it's 1. Different alleles result in different noses. We all have noses. We all have a nose gene, but different alleles are going to result in different varieties of noses. All right, let's divide everything up here. We're going to reform the nucleus here in each cell, and then we're going to divide it here. And remember, we're making male gametes here. And so these are spermatids, and then they're eventually going to mature into sperm, and they may or may not get used in reproduction, but they're down to one copy of each chromosome. So just to emphasize what can happen when we continue to look at randomness here, let's go back to Anaphase 1 here. Let's separate these. And notice it's light green, and dark green, light purple here. If we go back, we get dark, dark, light, light. So this is an opportunity here to get randomness here. How these chromosomes divide up creates more opportunity for variation in any particular individual. And in any particular individual, that translates to the species as a whole. The more diverse individuals you have, the more diverse the species is. And we'll talk more about that when we talk about evolution next semester. So let's continue. Let's click on each group of chromosomes here to reform them. We'll reform our chromosomes. We can drag a chromosome here and look at the allele map. When we've crossed over, you can see the capital letter stands for the dominant form of a gene. The dominant form of a gene always shows up no matter how many copies there are. The recessive form of the gene here, represented by the small letters here, that we crossed over with the other chromatid from the other parent, creates a new chromosome. There's a new combination of alleles here. We can bring over one of the purple chromatid here, and we can look at, we've got some crossover there as well. If we bring over the dark purple one, we can see that it was recessive, which isn't very common, but just for the sake of demonstration here, it had all recessive and it gained some dominant alleles from its the other parent. So let's drag the centrosomes over here. We're going to have another opportunity here to mix things up, because how things line up on the metaphase plate, whether the mixed up sides are on the same side or they're on opposite sides, that's another way to create variation or randomness. We're going to drag spindle fibers over, get everything nice and organized. If you don't do this, if you don't get the spindle fibers all nice and tidy, the cell might not divide correctly, and you might not have the right number of chromatids on each side. And then you can get a situation where chromosomal differences occur and the organism has problems. So we can go back and see if this turns out differently. Notice this is dark, dark, and these are both different, and these are light, light, and these are both different. Let's go back and see. Let's line these up and see if they line up the same here. They should line up differently, because that is a place, anytime you have a metaphase plate, is an opportunity for things to line up differently. We could skip this step. If we skipped the step where we pulled the chromatids apart, one cell would end up with too many, and that organism would be short. And here, where you can notice, this is female meiosis, where you get one larger cell of the four. The other polar bodies are recycled, and this matures to an egg, which is different than creating sperm, because all four mature. So just to show some place where things can go wrong, we can skip attaching these to the spindle fibers. We'll just say skip this step. And here you have a mix-up here, because this just has one chromosome, this has chromosome 1, and this just has chromosome 2. So you're missing chromosome 2 in these cells, and you're missing chromosome 1 here. They should have purple and green genetic material in both the cells. And you can see what happens if things go wrong. Let's attach things so you can kind of see the difference here. So everything can be neat and tidy when it divides. And there we go. We should get red and green in both situations, or excuse me, red and purple, or green and purple in both cells. So you can see there's green and purple here and green and purple here. It didn't go just green or just purple like it did when we didn't attach the spindle fibers. So anaphase II in meiosis is another place where these chromosomes are lined up on the metaphase plate. And if we decide to skip this step, we don't neatly divide those homologous chromatids right here. If we don't, if we just skip this, we get a whole mix of wonky DNA here that we don't want. There's a lot of opportunities for things in meiosis to go wrong. It's a complicated process. You can see that it has a lot of steps. And so let's go back and look at just some of the differences. So now that you've had an opportunity to see the meiosis simulation, let's just go over meiosis one more time and compare it to mitosis. So let's look at some differences and some similarities between meiosis and mitosis. We'll compare and contrast. Crossover happens meiosis, doesn't happen mitosis. The chromosomes don't mix in mitosis. In meiosis, you get 4 cells, each with 1 copy. Mitosis, you get 2 cells, each has 2 copies. Mitosis makes exact copies. Meiosis mixes things up here. It creates genetically unique chromosomes that are a different mix of alleles that occurred in either parent of the organism. It happens only in reproductive tissue. It's more complicated. There are more places for things to go wrong. It produces eggs and sperm, and it's not a cycle. Remember, it's a timeout. Yet you produce reproductive cells, and they have to find another reproductive cell before they can reproduce. So it's not a cycle. Mitosis, on the other hand, mitosis makes exact copies of the original cell, and those cells can produce exact copies. Mitosis happens in almost all body cells. It's less complicated. There's only 1 cytokinesis instead of 2. It produces just good old regular body cells like skin cells and eyeballs and liver and heart cells. It's a cycle because new cells can make more new cells. So those are the main differences between meiosis and mitosis. You can probably think of more. Thanks so much.